The next one is by Olaf Chisbo again, and the title is Do Reproductive Traits of Atlantico Really Differ in Population Maintained Under Exactly Similar Experimental Conditions? So Olaf, when you want.
Okay, me again, and this time with the co-author is Terje Svåsand from the same institute as me, the Institute of Marine Research. This time we will be talking about rare Northeast Arctic cod. You see the abbreviation in parentheses, and rare coastal cod, and wild-caught Northeast Arctic cod. Rare means that they have been raised from the egg stage to adults. And then we talk a little bit about the background and the potential importance of egg buoyancy and, in parentheses, egg size as a stock separation mechanism, how egg buoyancy is regulated, problems in measuring buoyancy, and that is an important issue. The actual reason for doing this research, we are using some old data and materials and method, and our expectation, and some results and conclusions. I was very lucky to work in Namibia for some years. I had a couple of students there, and there was some very important work taking place there in the Benguela system project. And just showing this to give you an impression of what we have in mind. And in this system, the anchovy and sardine is spawning very close to the shore here. But in this system here, there's an upwelling system. So the eggs is transported up here and into the so-called Ekman layer, and then they're advected offshore. But the fascinating thing here is that the larvae, they settle down and are transported back again into the coast, and here they are in nursery area. So they have developed a unique strategy to be maintained along the coast. And this work was mainly undertaken by Sundby and Stenvik and colleagues, and Anders Thorsen and myself were one of these colleagues then. There is other work showing that actually this local fjord cod in Norway are genetically different. It's a very fascinating thing. And so in a paper by Cianelli et al., they were stating that population structure of Atlantic cod is favored and maintained by a balance between water circulation and egg characteristics. And they found that the eggs basically were trapped inside the fjord. They were not flushed outside. So this is one of the fjords. Another very nice thing about buoyancy is that it's a fundamental mechanism. You have genetics on one side, but very little evidence on the physiology on the other side. But here is an evidence, and that is based on a comparison between Baltic cod and Northeast Arctic cod, and the first author was Thorsen, Anders Thorsen. The actual value here are not that very important, but the important thing is to see the relative amount of protein and free amino acids here in those two populations. And what you see here in the Baltic cod, you have a much higher relative proportion of free amino acids compared to protein, while it is opposite for the Northeast Arctic cod. So that means that here the protein is proteolized to a larger degree into free amino acids, and that is creating a lot of free amino acids inside the cell, and then you get water influx. So then you get a bigger egg with a much higher water content. So the water content here is something like 98%, but for the marine eggs it's something like 93%. So it's a huge difference. So these eggs here are much more buoyant. Another thing is, that buoyancy is maintained by the so-called sodium-potassium ATPase pump. And this is a study that we are doing in Bergen at the moment. We have a PhD student from South Korea. And here you have this is just an example, and here you have the ATP, and here is the mean specific gravity, or the buoyancy. A low mean specific gravity means more buoyant egg, and this is from the recruit spawn there on wild-caught Northeast Arctic cod, and you see that there's a link between amount of ADP and the buoyancy. It was not so clear for the repeat spawners, but we know that the energy content of the cell has influences on the buoyancy. But a question that we should ask is, what are we actually measuring? And I'm giving just picked this from the literature, showing to demonstrate what I'm talking about. And this is a study that was undertaken on Baltic Sea cod, and this was on 6 females. So here you have time, and here you have the egg-specific gravity or the buoyancy. So if you look more closely into these figures, you see this is one female, measuring the buoyancy, and here you see it's a breaking point, and then they start to be heavier. You see also here, goes down and heavier, here also heavier, here is some increasing. So you get a very big difference here. But maybe I'm a little bit biased, but my favorite is this one. And what you see special with this one is that it's continuous all the time. It doesn't have any breaking point. So if it's like this instead of like this, the model run will be very, very different. Okay, so I'm going a little bit into why we are doing this research. It's known that in Lofoten, Northeast Arctic cod and coastal cod, they are spawning at the same time, at the same place. And they are separated by a genetic marker, the pantophycine system. So this is the coastal cod, and this is the Northeast Arctic cod. And the institute is doing this genetic analysis. And this spring it has been a lot of discussion on this, and I can tell you why. And the situation that for the Northeast Arctic cod is on an all-time high. So if you look at the screen here is Northeast Arctic cod, and this is the spawning stock biomass. And you see here is we are now, and the spawning stock biomass is something like 1.2 million ton. While this is for coastal cod, and you must notice here that it's a different unit here. So this here has 100,000 tons, and this is 10,000 ton. And you see we have a steady decline in the coastal cod, and the spawning stock biomass is now something like 60,000 ton north of 62 degrees north. So it's a very big difference. So what is happening in Lofoten is that it's overflown by Northeast Arctic cod, while we are trying to protect this coastal cod where we don't want to have any fishing. Okay, so what is happening that we are introducing these closing areas in Henningsbergsboksen depending on this genetical analysis I showed you, and that has not always been popular among the fishermen, to say it in a very nice way. So sometimes it has been closed and then it has been open, okay? But then I ask myself, are these two stocks really different? The geneticians are saying that they are different, but is there any physiological evidence that they are really different? Okay, so this is the materials method. So we are using wild caught Northeast Arctic cod and reared Northeast Arctic cod and coastal cod. This is my favorite fish. They are very harmonic, say good morning to you. These are often a little bit neurotic, at least 1/3 of them. So I'm using this as a reference fish to quality check what is happening with this fish. So the good thing here is, of course, that you have the history of the fish. Here you don't know so much about the history. Okay. So we did this rearing of specimens in Parisvannet. It's northwest of Bergen. We kept them here in sea cages on this on-growth. And afterwards, they were transported to the spawning tank in Bergen. All 3 categories were there. And this is an example of a young student. And this is a teacher. And he's a member of the technical staff. And he was saying very much wise words at that time. And this is the circular tank. And also Fran was working there for some time. We have used it a lot also this time. So what is the expectations? Compared to Northeast Arctic cod, coastal cod should show higher egg dry weight content, and generally smaller eggs. That reason is that I should be deeper in the water, settle down earlier, and stay in the Lofoten area, while the other ones should be transported north for the northeast Arctic code. So the egg-specific gravity should also be higher, or buoyancy less for the coastal code, to avoid being transported northward with the current. Okay, is that really the case? So the first thing here we was looking was on the egg size, and this for 3 fish. So what is in blue, or degrees of blue here, is the northeast Arctic code from the circular tank, and what is in red and brownish here is the coastal code, and this is the egg diameter, and this is the portion of egg spawn. So 0 means that they have just started to spawn, and 100% is that they are spent. And that this looks very convincing. The eggs of the northeast Arctic code appears to be larger compared to the coastal code. But note here that we have only 3 fish of each category, so this can be, we can be lucky, to say it in a. So if you combine all the production for this from these 6 fish and 3 from each category, you see that the northeast Arctic cod, the rear northeast cod, have bigger eggs compared to the coastal cod. So that seems logical, what I was expecting. Okay. But then we plot the egg dry weight on the egg diameter, and you see that they are on the same, basically on the same line. That is not what I expected. I expected to find the coastal cod down here and the northeast Arctic cod up here, but they are on the same line. So this is a more robust test than the previous one. But please notice this fish here, which is 115K, that is a coastal cod. Okay. So then I went into the wild-caught northeast Arctic cod, which I know that is a very harmonic fish. And then you see that this egg dry weight content is the dry weight divided by the egg volume. And you see here, and this is the buoyancy or egg-specific gravity, and you see that's a very nice relationship between those two, indicating that you can be using the egg dry weight data to look at the buoyancy. But then you have 2 points up here. And the eggs look perfect, but what is happening here? Probably salt has gone into the cell, making that the specific gravity is too high, but we don't see it. So by using this here, we can have a test on the egg buoyancy data if they are trustworthy. Okay. And we did the same for the rear code data, and we found that we didn't have a problem. So we could not take out any buoyancy data. But this is from the gastrulation stage, and that made me a little bit worried, so I switched to a little bit earlier stage, and that is the so-called lens stage, which we believe is a more robust stage. So we used buoyancy data from lens stage on the rear code. And this is showing the results. So here is the rear coastal code, and here's the rear northeast Arctic code, mean egg diameter here, and buoyancy egg-specific gravity here. And what you see here is that there is a big overlap in the egg-specific gravity. One is down here, but if you go back to the fish I mentioned here, that is this fish, 115. So actually this egg dry weight data can be used to tell you something about the buoyancy. But this is opposite to expectation. They are lower in egg-specific gravity, while I was expecting that should be the coastal code should be up here, and the coastal code northeast Arctic code should be down here, but that is not what we see. So the conclusion is that measuring buoyancy of pelagic eggs is a straightforward job, but getting sensible data is another story. It is known that egg buoyancy is largely driven by water influx, proteolysis, and maintained by energetic nucleotides. Egg dry weight content data can be used to quality check the egg buoyancy data. Reared coastal cod apparently showed smaller eggs than reared northeast cod, but the egg dry weight versus egg diameter relationship were similar. Why I say apparently here is that we had only 3 in each category. So our conclusion is that there is no convincing evidence that the egg buoyancy of reared and coastal cod and northeast Arctic cod differ markedly. And then I end with an old photograph from Paris one where we undertook the studies.